learning objective 5, explain diffusion and osmosis. So we've already been talking about diffusion. Let's make sure that we're totally clear on what that means. So, passive transport is diffusion of a substance across a membrane with no energy investment. Diffusion, as you can see here, is the tendency for molecules to spread out evenly into an available space. And because the molecules are just going to move. They're always in motion. They're going to bump into each other, and so they're naturally just going to spread out until something impedes them from spreading out. And even when that happens, they're still going to be bumping into each other, but at equilibrium, you can't tell that they're moving anymore because your tea is not going to get any more brown. The particles in the solution will just keep bumping into each other. They're still moving, but it doesn't look like anything's happening because it's come to equilibrium. So at dynamic equilibrium, as many molecules cross the membrane in one direction as in the other. So here we've got a semi-permeable membrane. There's water on both sides, and say we've got these molecules of dye or tea or something like that that can move across the membrane. So because there's a higher concentration over here than there is on the right side, they're going to start to diffuse across that membrane. And the net diffusion is going to be from left to right. So some period of time passes. Some of those orange dots are going to get over to the right side. And then eventually, there's going to be as many of the orange dots on one side of the membrane as on the other. They're going to keep moving around, but the net rate of orange balls moving from left to right is going to be the same as them moving from right to left. And that's what we call equilibrium. Same concentration on either side of this membrane. What if we had two different solutes? Well, the same thing's going to happen in both directions. So if we start with orange on one side and purple on the other side, you can see even though the concentration of purple is lower than the total concentration of orange, there's going to be net diffusion in both directions. And at equilibrium, the concentration of orange is going to be the same on both sides, and the concentration of purple is going to be the same on both sides. So no matter what, there's going to be more orange on either side than purple. So more orange than purple on the left, more orange than purple on the right, but the concentration of orange is the same on left and right, and the concentration of purple is the same on left and right. So through diffusion, you could have all the solutes in here, and it could be very complicated, but at equilibrium, there's going to be equal proportions of each solute on either side of the membrane. Okay, so osmosis is diffusion of water across a membrane, a selectively permeable membrane. So water is going to diffuse from the region of lower solute concentration to the region of higher solute concentration until solute concentration is equal on both sides. So imagine in this example that these particles cannot cross that membrane, but water can. So water is going to eventually come to equilibrium by diluting whatever solute particles are on a particular side of that membrane. And water can even be made to do some work through osmosis. So here we've got a tube in the shape of a U, the letter U, and we've got two different concentrations of sugar, which is our solute, in the solution. We've got a selectively permeable membrane that doesn't allow the sugar to pass through, but it does allow the water to pass through. So what's going to happen is water is going to move from the area of lower concentration to the area of higher concentration. And after some period of time, you can see what's happened to the water in the tube. It's gotten lower on this side, it's gotten higher on this side. Why? Because osmosis is a force that's going to drive this this pair of reactions to equilibrium. So now, when the equilibrium has been reached, you can see the concentration of sugar on this side of the tube is about the same as the concentration on this right side of the tube. So not as concentrated over here anymore, because the water has made the concentration the same. Okay, so we need to learn a couple of new terms. Hypotonic, hypertonic, and isotonic. And what happens when a cell is placed into one of these types of solutions. Okay, so tonicity is the ability of a solution to cause a cell to gain or lose water. Because cells have a selectively permeable membrane, it's permeable to water. Now, if it's an isotonic solution inside and outside the cell, the solute concentration is going to be equal inside and outside the cell. And there's going to be no net water movement across the plasma membrane. Iso means the same. Remember, isotopes were different number of neutrons in the same element, isotopes. Isomers were the same formula of in a molecule, but different shapes, different arrangements of covalent bonds. So iso means same as. Okay, in a hypertonic solution, hyper means over and above, greater than. If someone is hyperactive, it means they're overly active. So that's how I remember that hyper means over and above, greater than. So if in a hypertonic solution, solute concentration is greater than outside the cell, than inside the cell, excuse me. And the cell is going to lose water through osmosis, just like in this tube here, the concentration, this one's hypertonic, this one's hypotonic. So this side is going to lose water. This water is going to go down in order to try and equal out the concentration on either side of the tube. In a hypotonic solution, hypo means below and beneath. A hypodermic needle goes under the skin. That's what it means, hypodermic. So that means below or beneath, less than. So in a hypotonic solution, solute concentration is less than inside the cell, and the cell is going to gain water. So let's go back to our U tube again. So at the start of this, we've got a hypotonic, lower concentration of solution on one side, and a hypertonic solution on the other side. So the same kind of thing is going to happen. But if we're talking about a cell which was put into a hypotonic solution, that means that the cell is going to gain water. Cell gains water. So the cells are affected differently depending on if you've got a plant or an animal cell. In an animal cell, if you put it into pure water, a red blood cell, into a hypotonic solution, water is going to go and try and dilute the contents of that cell so that it is equal on the inside and the out. And what's going to happen is that cell is going to burst because it can't hold all of that water. It's like trying to put too much air into a balloon. So lysis means breaking. You can see what's happening to this red blood cell. An isotonic solution, water goes in, water comes out. It's the same concentration inside and outside the cells. And so the net effect is nothing. Nothing happens to that cell in an isotonic solution. And that's what you want. If you are dehydrated and you need to get an intravenous fluid, you want the water that they're putting in to be isotonic with your red blood cells. So I often wondered, why do they give you a saline solution or a saltwater solution if you're dehydrated? And the answer is because they don't want your cells to burst. They want the saline solution that they give you is isotonic to your red blood cells. So water is not going to net shrivel up your cells or burst them. Okay, in a hypertonic solution, so imagine this is a very concentrated solution of salt. What's going to happen is it's going to pull water out of the cell and it's going to shrivel. So this is what you don't want. This is why you don't want to have hypertonic saline in your veins, because it's going to damage your red blood cells also by shriveling them up, by pulling water out. So if you're an organism that lives in a hypertonic or hypotonic environment, that can put you through an osmotic stress. So many organisms need to osmoregulate or balance the water that's in the cell with the water that's outside the cell. They need to do that actively. So a paramecium, which lives in a freshwater pond, is hypertonic. So since it's hypertonic, water is going to try and get in there and dilute the paramecium cell. Well, the paramecium certainly doesn't want to get diluted to death, so it's got a structure called a contractile vacuole that pumps fresh water out and keeps the cell intact. So that's a contractile vacuole, one type of vacuole. And you can see that these channels here are filling that contractile vacuole with water. And if you see this under a microscope, it'll sort of squeeze that contractile vacuole and all that fresh water goes back outside of the cell. All right, this is our last learning objective. If a dialysis bag containing a 1% solution were placed in a beaker containing a 5% solution, what would happen to that bag? Would it lose or gain weight? Is the solution in the bag hypotonic, hypertonic, or isotonic? Okay, so dialysis tubing is something that is used in hospitals and in dialysis centers. If your kidneys aren't working well, dialysis is a way to kind of artificially filter your blood. So you get hooked up to a dialysis machine and it does the job that your kidneys normally do for you, only you're hooked up to a big machine instead of having two nice kidneys doing that job for you. So dialysis tubing is a type of semi-permeable membrane, and we can use it for experiments to see what happens if we put hypertonic or hypotonic solutions inside the dialysis bag. So osmosis is going to move water from the area of high concentration to the area of low concentration. So what you can see in this picture here is that maybe we've got a protein that's inside our dialysis tubing, and that can't, and the membrane is not permeable to that. So the net effect on that molecule is nothing because it can't get outside. Okay, so maybe we've got a reagent that's represented by these green dots that is inside, and it's all inside the dialysis tubing. None of it is in here, but the dialysis tubing is permeable to those green dots. It's also permeable to these red dots, which they have labeled here as dialysate. And none of that starts out inside the tubing. But what's going to happen is, over time, the red dots are going to go inside the bag, the green dots are going to come outside the bag until the concentration comes to equilibrium. Okay, but say our solute can't get into or out of the bag. All it can get into or out of the bag is water. So if we have a 1% solution inside that dialysis bag, and we place that in a beaker with a 5% solution, then would the bag lose or gain weight? Okay, well, hopefully we can. Let's answer the second question first. Is the solution in the bag hypotonic, hypertonic, or isotonic? Well, I think we can rule out isotonic because 1 and 5 are not the same number. So there's less solution, less concentrated solution inside the bag. So the solution in the bag is below and beneath. It is hypotonic, not hypertonic. The solution in the beaker is hypertonic relative to what is in the bag. Okay, so now that we said that the solution in the bag is hypotonic, what's going to happen? Is the bag going to gain or lose weight? Give you a second to think about it. So what's going to happen? In a hypotonic solution, water is going to leave the bag, right? Let's go back and look at this. We've got a hypotonic solution. That's surrounding the dialysis bag, or in this case, the red blood cell. So whenever you have hypo and hypertonic, it's always relative to the other side of the membrane. So here, if we put a red blood cell into a hypertonic solution, the inside of the red blood cell is hypotonic. So what's going to happen? That red blood cell is going to try and dilute the more concentrated solution around it, and that's going to draw water out, and it's going to shrivel. Okay, so the solution in the bag being hypotonic means it's going to try and dilute the solution around it, so it's going to shrivel up. So hopefully you've been in lab and you've seen this in action and you understand it. It's difficult to explain, but it's a lot easier when you have the practical experience, which is why we have labs in the first place. Okay, so those are our learning objectives for Chapter 4. And up next, we're going to skip Chapter 5 for now. So skip right over that picture of Angelina Jolie in your textbook and go straight to Chapter 6, which is on metabolism and enzymes.